So, welcome to your third genetics lesson for Science 10. This is a continuation of section 1 on DNA, protein synthesis, and mutations. This lesson will focus on the DNA to protein component, so the protein synthesis part. So to begin with, just a reminder, you do have assigned readings for this course, and this is because you are not in class at all times. Although we have recordings of videos at home, it's not quite the same as being in a class consistently where we can kind of have discussions about these sort of things, at least not on a frequent basis. The next few slides here, which you are more than welcome to pause and read through, although the textbook will be linked for you, there are assigned readings for this. So this is just an overview of protein synthesis, starting off with transcription, and then going into translation. So we are going to talk about the process of protein synthesis in a bit more detail on the next few slides. Section 1.1, protein synthesis. This should all match the textbook, which you will have access to in class, and if I can post a digital copy. Let's go over this in a bit more detail. So part 1, the role of proteins. So again, this isn't referring to the protein that you eat. This is referring to protein within cells. So proteins are essential to life. They are made up of a packaged 3D chain of amino acids that form a specific shape for their specific function, much like a key is shaped for a specific lock. So if they're not shaped properly, they don't function in the way that they are supposed to, or they won't function at all. So that's where the analogy of the key and lock comes from. So the lock is a protein, which we call an enzyme in this scenario. And the key is what's called the substrate, or what fits inside of the protein or enzyme. So these two things work together to serve some kind of function within the cell. So proteins have two main functions, the first one being structural. So proteins help make up all structures in living things. So for example, muscle proteins, which you're most familiar with, hair, nails, and bones. This would also include things like fur, hooves, claws, or other parts of animals that are similar to hair and nails. And then also blood vessels and ligaments in the body. The second function is that they are functional, so meaning that they serve some kind of purpose within the body, or they have a role. So other proteins help us to keep our bodies functioning properly and to digest food. So enzymes, for example, speed up chemical reactions. Common example of an enzyme that people think of are the enzymes that help us to break down food. Hemoglobin in red blood cells, that's protein. Antibodies used in the immune system, so the body's ability to protect itself from viruses. This is antibodies, which block the receptor sites of viruses, very much like how these receptors are what in the media they're talking about the spike proteins on the COVID-19 virus. So this would be a receptor that could be blocked by an antibody. And then transport proteins found in the cell membrane. So in Science 8, you learned about how the membrane is semi-permeable, meaning that only some things get through. So it controls what goes in and what goes out of the cell. So these are proteins that are embedded in the membrane that actually allow this transport or semi-permeability to occur. So remember that DNA is a huge molecule, and that not all parts of that DNA code for a gene or code for a protein. So only certain genes are activated in a cell. So this is what makes one cell function different from another. Depending on which genes are active, different proteins are produced. And this causes cells to have different functions. So for example, you do not have skin pigment in genes being used in your stomach cells. So skin cells produce a protein called melanin, and your stomach cells don't produce this at all because it doesn't serve the function in your stomach. So one gene codes for one type of protein, which carries out one function in your body. The type of cell determines how those genes are actually activated. Okay, so for this slide, this is examples of different proteins throughout the body. So we've got things like antibodies, cell messengers, muscles, hair, nails, blood, brain and nerves, enzymes, and cellular construction workers. So what I would like you to do here is realize that different proteins are activated in different parts of the body, carrying out different functions. And each protein has its own name. You don't have to know all the different names, but this is just to help you realize the different functions and which proteins are involved in these different systems. So please pause here and take some time to just read through the slide so that you have an idea of the various proteins that are within your body. Okay, so part 2 of protein synthesis. So a reminder that the role of proteins is structural and functional. The process of making proteins is actually called protein synthesis. So let's break this process down a bit. There are 2 main steps for making a protein. So the first step is transcription. And this is where the DNA is transcribed or made into something called mRNA, which is short for messenger RNA. To transcribe simply means to copy something out. For example, you transcribe your notes while watching this video. So the copy is made in a slightly different form. So this is going from speaking to writing the words out. So they're still the same word, but they're just in a different form. The second stage of protein synthesis is something that's called translation. So this is where it takes that mRNA that was produced and translates it into an amino acid chain, or makes it into an amino acid chain. The best way to think of this is to think about translating a language, i.e. going from English to French. So you're taking something and turning it into something entirely different. So one language to another, where the words are entirely different from one another, although they mean the same thing. So let's go into this in a little bit more detail. So step 1, as we mentioned in the last slide, was transcription. And this is broken into the Latin terms trans, which means across, and scription, which means to write. The coded message of a gene on DNA has instructions on how to make particular proteins that our body needs. The instruction from a gene is copied from DNA to messenger ribonucleic acid, which is mRNA in the nucleus. So all of this transcription process is happening within the nucleus. So ribonucleic acid is RNA. Just like how deoxyribonucleic acid is DNA. And the M in the front of that RNA stands for messenger. It is important to know that this process of transcription occurs in the nucleus because DNA is unable to leave the nucleus. In DNA, we have the 4 bases, which we have already been talking about. So, and those are listed to the side below DNA. So adenine, thymine, guanine, cytosine, A, T, C, G. The mRNA pairs up with this, but it has one slight variation in its bases. So there is no thymine in mRNA. Instead, we have uracil. So that's shown here. And I'm just going to write out the word so you can see it. So uracil. So the mRNA code then that pairs with that DNA is UAGC. So there is a T in DNA and a U in RNA, which I just circled. Uracil is what pairs with the adenine in DNA, but adenine still pairs with thymine in DNA. So that's what you can see with those 2 rows. So take a minute, look at that, and make sure you've got that in your head. With the cytosine and guanine, the C and the G, nothing changes there because both are present in both DNA and RNA. Okay, so what we just covered in that last slide was that RNA has slightly different bases, A, C, G, and uracil, symbolized by the U, instead of thymine. So this is in DNA. Once complete, the mRNA moves through the nuclear pores and into the cytoplasm where proteins are made. The process of making mRNA, again, is called transcription. So our transcription step is simply to make mRNA, which can leave the nucleus and enter the cytoplasm, which is where proteins are made. So if we think about the Pfizer or Moderna COVID vaccine, this is an mRNA vaccine, meaning that it can be translated in the cytoplasm to make the spike proteins so that the cells in our body can recognize these, thus helping our immune system and developing antibodies against these spike proteins if you were to come in contact with the virus. So there is a simulation of this, which I will post, but I'm just going to talk you through these steps ahead of time so that you kind of understand what you're seeing. So step 1 in transcription is the unzipping process. So unzipping of DNA from a double strand into 2 single strands. So essentially the DNA is in 2 strands with hydrogen bonds in between the bases. So for example, where A pairs with T, that bond in between breaks. So the unzipping is these hydrogen bonds breaking, separating the 2 strands. Step 2, ribosomal bases pair up with the bases on one of the DNA strands to form mRNA. Step 3, enzymes help form the RNA backbone and checks for errors. So enzymes are another form of protein. They help form the mRNA backbone and they then check for errors. So the RNA backbone is still a phosphate and a sugar. So that's important to know. But this time, the sugar is different from DNA. So in DNA, the sugar is deoxyribose, but in RNA, it's ribose. So these two together form the backbone of the DNA. And it checks for errors just to make sure the copying was done correctly. Step 4, the mRNA is released, it leaves the nucleus, and the DNA zips back up to ensure that it's protected and ready for the next time transcription takes place. 1.2 protein synthesis transcription. So this is just an overview image slide to kind of show you what's happening. So here we see the DNA shown. And you can see at the ends of the strands here that the DNA is still in the form of a double helix on the edges. But that center part that forms kind of a loop structure is 2 separate strands. So this is the unzipping process. So for example, A breaking away from T. If we look at step 2 and 3, the ribonucleotide of RNA, which is just the base nucleotide of RNA, comes in and pairs with the single DNA strand, forming our new mRNA strand. This pairing only occurs in this one section where the gene is, not through the entire length of DNA. So that's why you can see that some parts of it are still zipped up. And then in step 4, the mRNA leaves the nucleus and enters the cytoplasm. So to kind of give you an overview of this whole process, the nuclear DNA pairs and forms mRNA. That mRNA leaves the nucleus through nuclear pores and migrates to the cytoplasm where proteins are actually made. All right, so at the bottom here, just like getting you to go through the DNA replication process or the DNA pairing process, where you're pairing the DNA bases, we're now going to do that with one strand of DNA pairing with the complementary mRNA strand. So I'll go through one example with you here, and then I would like you to pause and try the last two on your own. So after this, you are then going to look at the simulation that I mentioned, at least the first part of it, for a bit more practice, and then you can go through the next set of notes. So a reminder, A pairs with U in RNA, but T in DNA still pairs with A. C and G pair with each other just as they would for DNA. So our first strand here then is TAC. T pairs with A pairs with U, C pairs with G, T pairs with A, C pairs with G, G pairs with C, and then G pairs with C, C pairs with G, A pairs with U, C pairs with G, A pairs with U, G pairs with C. So that is the complementary strands. You can see that there's little spaces between the three letters. This is just to help you see it a little bit better. It does serve a purpose with protein synthesis, which we will potentially get into later, although this is more of a bio 11 and 12 sort of thing. Also check the DNA versus RNA comparison sheet on page 13 of your notes. Look over the diagrams and look at the similarities and differences between DNA and RNA in terms of its structure. Look at the sugars. How are the sugars different? Can it leave the nucleus when you compare the two of them? And then look at the type of the molecule. Okay, that is it for this portion of the lesson. So after this, and once you've done your simulation, you can move on to the next video.